Predictor?
Yes,
I
AdenPredictor.
thought you're gonna read it, right?
Yeah. Okay. Hello, everyone. Big hugs. Thanks for coming to my talk. If it wasn't for you, this room would be empty. So my name is Mihir, and I'm going to present some work that was done in conjunction with Roma Baral, who used to be a master student in our lab. Abhinav Aduri, who's in the audience. He's a final year PhD student. Dong Huiyan, who's not here, but also a final year PhD student. And Hussein Mohammani, who's our advisor at Carnegie Mellon. And the title of my talk is AdenPredictor, Accurate Prediction of the Adenylation Domain Specificity of Nonribosomal Peptide, Biosynthetic Gene Clusters, and Microbial Genomes. I admit, this title is very long, and it's also probably the case that most people in the audience are not really familiar with any of these words. So, succinctly, what this talk is about using machine learning to discover non-ribosomal peptides. Some examples of non-ribosomal peptides are penicillin, romidepsin, and echinocandin. Each one of these is approved by the FDA to sell to the American public. Now, there are many, many other non-ribosomal peptides that are approved by the FDA, and there's many, many more non-ribosomal peptides that are in various phases of the clinical approval process. Now, you may be asking, why use the word non-ribosomal? And so penicillin is an antibiotic, romidepsin is an anti-cancer molecule, and echinocandin is an antifungal. Now, why should we use the term non-ribosomal? So in the 60s, scientists were studying a bacteria known as Bacillus brevis. They wanted to inhibit all peptide and protein production. So they laced the cell plate with ribosome targeting antibiotics. Despite this, some peptides were produced, namely tyrosine. So somehow tyrosine did not depend on the ribosome, and thus the word non-ribosomal was coined. Over the subsequent years, scientists developed an overarching picture of what's going on. There's an enzyme known as non-ribosomal peptide synthetase. It has many regions called A domains, and they work together in a giant molecular assembly line. First, the first A domain, A1, will recruit an amino acid and then pass it on to A2. A2 will add an amino acid to that, pass it on to A3, and so on. Over the course of this assembly line, the molecule is rarely linear. Often you'll see some cycles and you'll see some branches. Now, given this overarching structure, the last kind of step to figure out which molecules are generated by these enzymes is to figure out which amino acids are recruited by each A domain. Scientists examined these A domains and for the most part found that most of the regions were quite similar. There were a few, though, that were quite variable. And so, in 1999, a scientist known as Stachelhaus et al. Decided there's an 8 amino acid code where there's a lot of variability, and he thought that probably these 8 amino acids are deterministic of which amino acids are recruited. Later on, in 2009, Rausch et al. Expanded this code to 34 amino acids. Now, 34 amino acids is hard for a human to visualize, so then he said, okay, well, let's use machine learning models that can handle high-dimensional data sets to predict which amino acids are recruited by which A domain. Then lastly, in 2011, Rodig et al. Developed NRPS Predictor 2. Now this model diverged from previous models in two distinct ways. First, this model used both unlabeled data and labeled data. In this domain, there's only maybe 1,000 to 2,000 labeled A domains. Well, there's quite a bit of unlabeled data. So Rodig et al. Decided to use a semi-supervised learning algorithm to improve the accuracy. And number two, he thought that physiochemical properties of each amino acid could be quite predictive of which amino acids are recruited. And so each amino acid was mapped to a 12-dimensional vector. This vector contained information about hydrophobicity, electron properties, and size. Now, this model is now considered state-of-the-art in the field. People working in this area all know about NRPS Predictor 2. Now, you'd be asking, okay, state-of-the-art. So can we now just predict the structure of the non-ribosomal peptides given the enzyme? The truth is it's not that easy. So first of all, NRPs have extensive modifications. So even if you knew all the amino acids recruited, the molecule is still modified in several places. Number two, NRPS predictors choose top K accuracies high. Top K meaning for any input, you output the top 3 or 4 entries. And if it contains the true label, then you have a hit. If it doesn't, then you got a not hit. Its top K accuracy is high, but its top one is not. And NRPs are not linear. Cycles are formed in various positions. Because of these kind of like multiple options, if you were to only generate one molecule, it's very unlikely you would guess the true molecule generated by the enzyme. Thus, if you want to capture the real molecule produced, you have to guess probably 10,000 to 100,000 molecules to make sure you would have guessed, or in that set, you actually have the true molecule. So, in 2021, Bahar Behzaz, who's now a postdoc in our lab, but did her PhD with Pavel Pevsner, she developed this tool called NRP Miner. What it did is first generate a large set of molecules that she thought probably contained the real molecule generated by a non-ribosomal peptide synthetase enzyme. And then she would confirm which molecules actually exist in sample using mass spectrometry. And this worked in the following way. For each hypothetical structure in the set, she would predict the theoretical mass spectra, meaning if the hypothetical structure went through a mass spectrometer, what mass spectra do you expect to be output? Then NRP Miner would recruit a bunch of real mass spectrometry data. For each experimental mass spectra, it would compare it against the theoretical mass spectra. If any of the experimental mass spectra are close to the theoretical mass spectra, then it would be believed that the hypothetical structure that corresponds to that theoretical spectra exists in sample. Great. So NRP Miner was developed. It discovered many new non-ribosomal peptides, but for some reason it failed to discover some known molecules we knew are generated by some microbes. We examined the code, the several modules in the code, and we found that NRPS Predictor 2 was failing. Interesting. State of the art, but failing. So we decided to go examine NRPS Predictor 2. Okay, here is a graph. In blue is shown the top 3 accuracy of NRPS Predictor 2. In gold is shown top 1. The top 3 accuracy is quite good. It's above 90%. So how could NRPS Predictor 2 be failing? Doesn't really make sense. So we examined the A domains involved in the creation of molecules that we knew should be generated. And we found out that the A domains were actually far away from the training data with respect to the Hamming distance. Because of this finding, we decided to divide our test data into buckets. Buckets would have varying degrees of difference between the test data and the training data. BK plus here means all test data points that are different than training data and at least K amino acids. So if there's a test data point that is different than a training point in 3 amino acids, it will never belong to B5 plus or B6 plus because it's close to a training data point. It could belong to B1 plus, B2 plus, or B3 plus. And then we decide to see what is the accuracy, at least top 1 accuracy, of NRPS Predictor 2 for varying buckets. Okay, now here we can notice 2 things that are very interesting. Here's the overall accuracy on the left. For B6 plus, the accuracy drops dramatically. What this implies is that a lot of the great accuracy for overall is due to test data points that are really close to training data. But NRPS Predictor 2 is failing for things that are far away. Namely, for B21 plus, NRPS Predictor 2 is abysmal. So, even though NRPS Predictor 2 test accuracy was quite good, it didn't mean the model generalized. Interesting. Okay, great. So then the question was, initially, how do we build more generalizable models? And everyone who does machine learning knows the standard stuff. You maybe try better feature vectors, you try other ML algorithms, and just the reason we're trying to get better feature vectors is we have limited data. In the case of deep learning, they call it representation learning. You can learn your own representations, but you only have 1,000 labeled points. You kind of have to posit some better feature vectors. So we were stumbling around. Like I said earlier, NRPS Predictor 2 uses a 12-dimensional vector that corresponds to an amino acid's hydrophobicity, size, and electron properties. And for each of the 34 amino acids in the non-ribosomal code, it concatenates all these 12-dimensional vectors and uses that input. And just to be sure, we thought, okay, let's try one-hot encoding. Let's make sure that physiochemical is indeed really beneficial. So we did this, right? So for each amino acid, we mapped it to a one-hot binary vector, meaning that the first amino acid is mapped to a vector with a bunch of zeros, except 1, 1 in the first entry. the second amino acid, a 1 in the second entry, and so on. And we noticed that physiochemical wasn't that much worse or better than one-hot encoding, which was surprising given the previous paper was kind of mentioning this as an important step. And then we also tried some other ML algorithms, right? To make it aesthetically pleasing, I've just shown 2 others besides NRPS Predictor 2. We tried a deep learning method, graph neural networks. They did worse than NRPS Predictor 2 on the overall accuracy, did significantly better than NRPS Predictor 2 for B21+. The extra tree algorithm, basically kicked butt. It did better than NRPS Predictor 2 on almost every bucket. Cool. So what we noticed, though, however, is that we still have this issue, right? Your accuracy is still dropping as your bucket numbers go higher. So let me ask the question, what's another approach to build more generalizable models? The current issue, as I mentioned earlier, is that a large fraction of the data is close to each other, right? In that plot where I showed buckets and the accuracy dropping dramatically between overall and B6+, right? A lot of the data was 1, 2, 3, or 4, or 5 Hamming distance away from the training data. And that's why the model was doing well. So the question is, how can you build a more diverse data set? The idea is to sample more varied A domains. And so we came up with an idea of how to do this as a proof of concept. What we do is mine the microbial genomes, cluster them with some standard clustering algorithm, and then check if any of these clusters contained A domains for which we know the recruited amino acid or labeled A domain. Here's a graph. Each point in the above and below graph corresponds to an A domain in low dimensional space. And the above graph points are colored the same if they belong to the same cluster. And the below graph points are given a color if we know the amino acid they recruit. Now, if you examine these graphs carefully, you can find some regions that are interesting. So in the top graph, you see 3 various shades of violet clusters. If you look in the below graph, though, there is no colored point, meaning none of these clusters have representatives for which we know the A domain. We found 50 such clusters. We haven't taken it all the way to the extent of actually examining the A domain. That would be expensive, but this is just a proof of concept. Nevertheless, we still have this kind of big bottleneck. We would like to build more and more generalizable models. So Abhinav Aduri in the audience had this really interesting idea. Why don't we take the folding of each A domain using AlphaFold, and then use molecular docking methods to see which substrates would bind to the binding pocket. The advantage of this approach is twofold. First of all, these molecular docking methods are trained on lots of data. In our domain, there's only 1,000 labeled data points. But if molecular docking is trained on a bunch of other data, maybe you can build a more generalizable model. And number 2, molecular docking uses physics-based principles. Over the past 50 years, some of the most successful deep learning models that deal with molecules have integrated physical symmetries such as equivariance. This work will be done with David Koh's lab at University of Pittsburgh. They're experts in computational tools related to folding and substrate binding. Great, so that's kind of the scope of what we've done in the paper and what we plan to do. Now I wanted to give a short recap of the lessons we learned. 2 of them I think are quite generalizable to all of computational biology, and 2 of them are quite more narrow to our domain. So NRPS Predictor 2 was accurate for sure. It trained on training data, tested on test data, and it had good accuracy. But that doesn't really correlate to generalizability. Because if a large fraction of your data is close to each other, right, a lot of your accuracy will due to the fact that your test data is close to your training data. And so even if you have high accuracy, it doesn't mean you're generalizable. So papers in computational biology, if possible, should do the following. You should divide your test data into buckets by measuring various degrees of difference between the test data and the training data. And the only way you're going to know if your model is truly generalizable is to look at the higher buckets. So that's something I think almost everyone in computational biology should start doing, if possible. In the case of images, it's hard to measure distance from training data. But in the case of our domain, we just cared about, Hamming distance in amino acid code. So it's quite possible. And then for our paper specifically, for some reason, one-hot coding did almost equally as well as physiochemical. The extra tree algorithm was the most performant model in the test data, far from training. This is interesting because like when you, well, I don't know, at least in America, no one ever talks about the extra tree algorithm. But this is the one that worked really, really well. It worked better than everything else. And so it's interesting that this is not kind of mentioned in the standard ML toolkit. And then lastly, we proposed a way to kind of build a more diverse data set. Although, of course, it's quite expensive. But at least you have a proof of concept. And yeah, that's the end of my talk.
Hey, I think we have time for a few questions. So I'll start one.
Yes, sir.
So have you tried ensemble approach? Because you tried several different way,
Yeah.
and if their mistake don't overlap, you may be able to get
You can get
better
better
performance. Just run all of those.
Yeah, we didn't try it. Oh well, actually, we did try it to be honest. We didn't seem to get that much better performance. Maybe if we did a weighted ensemble or something,
Okay.
but we didn't fully explore it. It's a
Okay.
good point. It's worth
Yeah.
doing more exhaustive search of it.
Yeah. So like normally, like my understanding for if your model does not generalize, normally that means your testing data is from a different distribution of your training data. So
yeah,
I was wondering, since I think the bucketing approach is a very good approach, but can you stratify when you splitting your training and testing? Don't use random split.
Yeah, you can try to use like the more variable data set if possible.
Yes.
Yeah, there's that. Yeah, I think that's definitely worth exploring. Yeah, I agree. But I mean, I totally agree. And then I just want to re-say like, it's interesting, right? Because you get a lot of good results, and in biology we hope that these models are truly generalizing other things.
Right. Yeah. Okay. Great. So.
A very interesting talk. Do you know why one-hot encoding works better than physicochemical? Any technical reason?
That's like the thing with machine learning. No one knows anything. Sometimes just, I mean, that's like just the truth. People, there's no real principles in machine learning or deep learning, and that's why you try everything. And so it's kind of unsatisfying intellectually, but such is life, I guess.
Okay.
I mean, what I would say if someone asked me, I'd be like, okay, well, the data is kind of tabular. On tabular data, kind of random forest tree-based models work fine. That's what I would say as a fake answer, but it's not really real. Yeah.
I think is a little bit ahead of time because I think your talk is 30 minutes.
Yeah. Sorry, I talked too fast. I apologize.
All right.
We're 20 minutes
there are
in.
more questions.
Okay, cool.
Thank you very much for your talk. My question is, so this method, is it already public? Can we try
Yeah,
it out?
it's we have a GitHub where it's on there. Some people have already started downloading and using it.
Okay.
So yeah, maybe you can, if you want to get access to it, you can talk to me and enough afterwards, and we can help you
perfect.
out. Yeah.
I saw one more question. So much easier to walk.
Yeah, thank you for the great talk. So I have a question also a bit related to one-hot encoding.
Yes, sir.
So one problem with the public data on ADAMA is that it is highly imbalanced, right? So you have like standard amino acids, for example, much more represented than non-standard ones.
Yeah.
So did you experience something like that when you tried this?
Yeah. So actually in our paper, I haven't mentioned this, but we did try weight balancing approaches. So we would change the loss so that each kind of label gets the same amount of loss to the whole kind of model. And it didn't seem to affect our results that much.
Okay.
Yeah, I mean, the extra tree algorithm performed a little bit better, like maybe like 0.2 or 3%, but we did try, and we thought, phenylene label occurs a lot, like 20% of the data, but it didn't really affect our results when we included weight balancing.
Yeah, okay, thank you.